So here you are, lying in the dark. Maybe your eyes are already closed. Maybe you're still settling in, adjusting the pillow, pulling the blanket up just a little higher. And I want to tell you about something that's happening right now, in this exact moment, inside you. Not metaphorically, literally. Right now, as you breathe in, oxygen molecules are slipping through the thin walls of tiny air sacs in your lungs, crossing into your blood, getting scooped up by red blood cells, and carried off to trillions of destinations throughout your body. Each of those destinations is a cell. And inside each of those cells, that oxygen is being used in a process so elegant, so intricate, that it took humanity centuries just to figure out it was happening at all. You don't feel it. You're not aware of it. But it's the reason you're alive right now. The reason you can hear these words. The reason your brain is doing the quiet, beautiful work of turning sound into meaning. Cells. Everything about you, your thoughts, your heartbeat, the warmth of your skin, the fact that a cut on your finger will heal in a few days, all of it comes down to cells. And here's what I love about this subject. Cells are simultaneously the most ordinary and the most extraordinary things in the universe. Ordinary because they're everywhere, because you're made of roughly 37 trillion of them, because every living thing you've ever seen or touched or eaten is built from them. And extraordinary because each one is, in a very real sense, more complex than any machine humans have ever built. More complex than a smartphone, more complex than a jet engine, more complex than the International Space Station. That's not an exaggeration. When you actually look at what's going on inside a single cell, the thousands of chemical reactions happening simultaneously, the molecular machines assembling proteins, the DNA being read and copied and repaired, it's staggering. And we're going to walk through all of it, slowly, gently, one piece at a time. But let's start at the beginning. Not the beginning of cells. They've been around for about 3.5 billion years, and we'll get to that. But the beginning of us knowing about them. Because for most of human history, nobody had any idea cells existed. And why would they? You can't see them. The smallest cells are maybe 1,000th of a millimeter across. Your eye can't resolve anything much smaller than about 1/10 of a millimeter, so cells are literally invisible to you. For thousands of years, people looked at flesh, at leaves, at blood, and saw continuous stuff. Smooth, unbroken material. The idea that everything alive was actually made of tiny, discrete, individual units. That idea required a tool that didn't exist until the 17th century. The tool, of course, was the microscope. And the story of how we discovered cells is really the story of a handful of curious people who pointed these new instruments at things nobody had ever really looked at before. The first person to use the word cell in this context was Robert Hooke, an Englishman, and he did it in 1665. Now, Hooke is one of those figures in the history of science who deserves to be far more famous than he is. He was brilliant, an architect, a physicist, a biologist, an inventor. He helped rebuild London after the Great Fire. He came up with ideas about gravity that later caused a bitter, decades-long feud with Isaac Newton. Newton, by the way, essentially tried to erase Hooke from history after Hooke died, which is partly why you might not have heard of him. But that's a whole other story. What matters for us tonight is what Hooke did with a microscope. He had built one himself, a compound microscope, meaning it used two lenses, one to gather light and one to magnify the image. It wasn't great by modern standards, maybe 30 or 40 times magnification. but it was enough. Hooke looked at all sorts of things: the edge of a razor, the surface of a nettle leaf, a flea, a louse. He published his observations in a book called Microscopic, which is genuinely one of the most beautiful scientific books ever produced. The illustrations are stunning. These enormous, detailed engravings of tiny things blown up to fill an entire page. The flea illustration alone folds out to about 18 inches wide. People were astonished. Samuel Pepys, the famous diarist, called it the most ingenious book he'd ever read. But the observation that matters most came when Hooke sliced a thin piece of cork, the bark of a cork oak tree, and put it under his microscope. And what he saw was a pattern: tiny, regular, rectangular boxes packed together like a honeycomb. They reminded him of the small rooms that monks lived in, which were called cells. So he called them cells. It's a word that stuck, obviously, though what Hooke was actually seeing wasn't quite what we mean by cells today. Cork is dead tissue. What he was looking at were the empty cell walls left behind after the living contents had dried up and disappeared. He was seeing the architecture, the skeleton, but not the living thing itself. Still, it was the first glimpse, the first hint that living matter had structure at a scale too small to see with the naked eye. Now, around the same time, and this is one of those wonderful coincidences in the history of science, there was a man in the Netherlands doing something even more remarkable. His name was Antony van Leeuwenhoek, and he was not a scientist, not in any formal sense. He was a draper, a fabric merchant in the city of Delft. He had no university education. He didn't read Latin, which was the language of science at the time, but he had an obsession with grinding lenses, and he was extraordinarily good at it. Leeuwenhoek made simple microscopes, just a single, tiny, perfectly ground lens mounted in a small metal plate. You held the whole thing up to your eye, almost like a magnifying glass. But because his lenses were so precisely shaped, they could magnify things up to about 270 times. That's far better than what Hooke's compound microscope could do. And with these little handheld instruments, Leeuwenhoek saw things no human being had ever seen before. He looked at a drop of pond water and found it teeming with life. Tiny creatures swimming, spinning, darting around. He called them animalcules, little animals. We now know these were protists, single-celled organisms like Paramecia and Vorticella. He looked at scrapings from his own teeth and saw bacteria. He was the first human being to ever see bacteria. Think about that for a second. For the entire history of our species up to that point, bacteria had been invisible, unknown, and yet they were everywhere, in every mouth, in every handful of soil, in every drop of water. Leeuwenhoek saw them in the 1670s, and he wrote letters about his discoveries to the Royal Society in London, and people could barely believe it. Some didn't. It took a delegation from the Royal Society visiting Delft and looking through his microscopes themselves before the skeptics were convinced. Here's the thing about Leeuwenhoek, though. He was secretive about his lens-grinding techniques. He made over 500 microscopes in his lifetime, but he never told anyone exactly how he achieved such high-quality lenses. When he died in 1723, at the age of 90, his methods largely died with him. It took over a century before microscope technology caught up to what he had been able to do with his simple, single-lens instruments, which means there was this strange gap in the history of cell biology. Leeuwenhoek had opened a window into the microscopic world, and then, in a sense, the window partially closed again. It wasn't until the early 19th century that microscopes improved enough and that enough people were using them systematically for the next big leap to happen. And this is where the story gets really interesting, because it's where we move from just observing cells to understanding what they mean. In the 1830s, a German botanist named Matthias Schleiden was studying plant tissues under the microscope, and he became convinced that every part of a plant was made up of cells or derived from cells. Every leaf, every stem, every root, every petal, cells all the way through. Around the same time, his friend Theodor Schwann, a zoologist, was finding the same thing in animal tissues. Muscle, nerve, cartilage, blood, all made of cells. The two of them got together, and in 1838 and 1839, they published what became known as cell theory. The idea, stated simply, is this: all living things are made of cells. That's it. That's the whole theory, at least in its original form. And it might sound obvious to you now, but at the time, it was revolutionary. It unified biology. It said that plants and animals, despite looking completely different, despite functioning in completely different ways, were built from the same fundamental unit: a cell. Now, there was a piece missing from Schleiden and Schwann's version of cell theory, and it's an important one. They knew everything was made of cells, but where did new cells come from? Schleiden thought cells could crystallize out of a formless substance, sort of spontaneously assembling the way crystals grow in a mineral solution. He was wrong about that. The correct answer came from a German physician named Rudolf Virchow, who in 1855 published a famous Latin phrase, omnis cellula, e cellula. Every cell comes from a cell. New cells are produced by the division of existing cells. They don't spontaneously appear. They don't crystallize. They divide. A cell grows, duplicates its contents, and splits in two. That's how you went from a single fertilized egg to the 37 trillion cells you're made of now. Every one of those cells is a descendant, through an unbroken chain of cell divisions, of that original single cell. And if you trace it back further, that original cell was descended from your parents' cells, and theirs from their parents, and on, back through an unbroken chain of cell divisions that stretches all the way to the very first cells on Earth, somewhere around 3.5 to 4 billion years ago. You are connected, through an uninterrupted lineage of dividing cells, to the origin of life itself. That's not poetry. That's literally true. So by the middle of the 19th century, scientists had the basic framework. All life is made of cells. All cells come from other cells. But they still had almost no idea what was going on inside a cell. They could see that cells had an outer boundary, a membrane of some kind. Many cells seemed to have a dark spot inside them, which they called the nucleus, from the Latin word for kernel or nut. But the interior of the cell was mostly a mystery. They called the stuff inside the cell protoplasm, which is a wonderfully vague word that basically means first formed stuff. It was a way of naming their ignorance. They knew something was in there. They could see it was sort of gel-like, slightly granular, clearly doing something important. But what exactly? The answers would come slowly over the next 150 years as new tools and new techniques peeled back layer after layer of cellular complexity. And what they found was astonishing. Inside that tiny space, a typical human cell is maybe 10 to 30 micrometers across, which means you could line up maybe 1,000 of them across your thumbnail. Inside that tiny space, there is a world of organized, purposeful molecular machinery that makes anything humans have engineered look crude by comparison. There are power plants that convert fuel into energy. There are factories that read genetic instructions and build proteins to order. There are transportation networks that ship cargo from one part of the cell to another inside membrane-wrapped packages. There are recycling centers that break down worn-out parts and reuse the components. There's a skeleton, a dynamic, constantly remodeling skeleton that gives the cell its shape and allows it to move. And at the center of it all, locked away in the nucleus like a master blueprint in a vault, is the DNA, the molecule that contains every instruction the cell needs to build and maintain itself. We're going to explore all of these systems one by one in the hours ahead. We'll talk about how the membrane works, how it decides what gets in and what stays out. We'll talk about mitochondria, those famous powerhouses, and how they may have once been free-living bacteria that got swallowed up by a larger cell in one of the most consequential mergers in the history of life. We'll talk about how DNA is read, how proteins are made, how cells communicate with each other, how they divide, how they die, and we'll talk about what happens when things go wrong, when a cell's machinery breaks down, when mutations accumulate, when a cell starts dividing out of control and becomes cancer. But for now, let's stay here for a moment, in the 19th century, because there's something happening in the 1880s and 1890s that's going to change everything. Scientists are getting better at staining cells, using chemical dyes to color specific parts of the cell so they show up more clearly under the microscope. And one of the things they're starting to see during cell division is something truly strange. The material inside the nucleus, which normally looks like a diffuse, tangled mess, starts to condense. It organizes itself into distinct, rod-shaped structures. They absorb certain dyes very strongly, so they're easy to see. A German anatomist named Heinrich Wilhelm Waldeyer gives them a name in 1888. Chromosomes, from the Greek words for color and body. Colored bodies. And as scientists watch cell after cell divide, they notice something remarkable about these chromosomes. They behave with extraordinary precision. They line up in the middle of the cell, they duplicate, and then they separate, with exactly half going to each new daughter cell, every single time. It's like watching a choreographed dance, and it's so precise, so orderly, that it's clear this isn't random. Something about these chromosomes is deeply important. They're being handled with care because they carry something the cell cannot afford to lose. And what they carry, though nobody quite realizes it yet in the 1880s, is the hereditary information itself. The instructions for building a living thing passed from parent to offspring, generation after generation. The blueprint, if you want to call it that. Though, as we'll see, it's really more like a recipe, a set of step-by-step instructions that the cell follows to construct everything it needs. Now, to understand how scientists figured this out, we need to talk about a quiet Augustinian friar working in a monastery garden in what is now the Czech Republic. His name was Gregor Mendel, and his story is one of those heartbreaking tales in science where someone does absolutely brilliant work, publishes it, and then watches it disappear into near total obscurity for decades. Mendel was born in 1822 in a farming family, and he entered the monastery partly because it gave him access to education he couldn't otherwise afford. He studied physics and mathematics in Vienna, and when he came back to the monastery in Brno, he started experimenting with pea plants. Not because he was trying to discover the laws of heredity, or maybe he was, actually. The man was clearly thinking big. But because he wanted to understand how traits get passed from one generation to the next. And here's what made Mendel different from everyone else who had tried to study inheritance before him. He counted things. He was meticulous. He tracked thousands of individual plants over multiple generations, carefully recording which ones were tall and which were short, which had green peas and which had yellow, which had smooth seeds and which had wrinkled. 7 traits in total. And he cross-pollinated them by hand, controlling exactly which plant bred with which. This was painstaking, tedious, years-long work. We're talking about the better part of a decade, from roughly 1856 to 1863, spent in a monastery garden with tweezers and paintbrushes, manually transferring pollen between pea flowers. And what he found was stunning, though almost nobody realized it at the time. Traits didn't blend. That was the prevailing idea. That inheritance worked like mixing paint, where a tall parent and a short parent would produce a medium-height offspring. But Mendel showed that wasn't what happened at all. When he crossed tall pea plants with short ones, the first generation was all tall. Not medium, all tall. But then, when he crossed those tall offspring with each other, something remarkable happened in the next generation. About 3/4 were tall, and 1/4 were short. The shortness hadn't disappeared. It had been hiding. Mendel called this the principle of dominance. The tall trait was dominant. The short trait was recessive. And that 3-to-1 ratio kept showing up, trait after trait, generation after generation. It was mathematical. It was predictable. It was, in essence, the first glimpse of a discrete, particulate theory of inheritance. The idea that hereditary information comes in distinct units that don't blend, but instead get shuffled and dealt out like cards. Mendel published his results in 1866 in the Proceedings of the Natural History Society of Brno. And then, nothing. Almost nobody read it. Almost nobody cited it. Charles Darwin, who was working on his own ideas about inheritance and would have benefited enormously from Mendel's work, apparently never saw it. Mendel became abbot of his monastery, got bogged down in administrative duties and a tax dispute with the government, and died in 1884 without ever knowing that he had laid the foundation for an entire science. It's one of the saddest stories in biology, honestly. The man figured out something profound, and the world just shrugged. But here's the thing. The world caught up. In 1900, 3 different botanists, working independently in 3 different countries, rediscovered Mendel's laws. Hugo de Vries in the Netherlands, Karl Correns in Germany, and Erich von Tschermak in Austria, all arrived at similar conclusions through their own plant breeding experiments. And when they searched the literature, they found Mendel had beaten them by 34 years. Suddenly, Mendel was famous. Suddenly, everyone was talking about these discrete hereditary units, which would eventually be called genes, a term coined by the Danish botanist Wilhelm Johansen in 1909. And now the question became, where are these genes physically? Where do they live inside the cell? And this is where those chromosomes come back into the picture. Remember those precisely choreographed rod-shaped structures that scientists had been watching during cell division? A few people started putting 2 and 2 together. If Mendel's hereditary factors come in pairs, 1 from each parent, and if chromosomes also come in pairs, well, that's a pretty suggestive coincidence, isn't it? The person who really nailed this connection was a young graduate student at Columbia University named Walter Sutton. In 1902, Sutton was studying grasshopper cells. Grasshoppers happen to have big, easy-to-see chromosomes, and he published a paper arguing that Mendel's factors of inheritance were physically located on chromosomes. A German biologist named Theodor Boveri arrived at essentially the same conclusion around the same time, working with sea urchin embryos. So this idea is often called the Sutton-Boveri chromosome theory of inheritance, and it was controversial. Not everyone bought it right away. The evidence was suggestive but circumstantial. What was needed was proof, a direct demonstration that a specific gene lived on a specific chromosome. That proof came from a tiny, annoying insect, and a man who initially set out to disprove the chromosome theory. Thomas Hunt Morgan was a skeptic. He was a professor at Columbia University, and in the early 1900s, he wasn't convinced that chromosomes carried hereditary information, so he decided to test it, and his experimental organism of choice was Drosophila melanogaster, the common fruit fly. You know those little flies that hover around overripe bananas on your kitchen counter. Morgan chose them because they're cheap, they're small, they breed fast, a new generation every 2 weeks, and they only have 4 pairs of chromosomes, which keeps things relatively simple. Morgan and his students crammed into a small lab at Columbia that became known as the Fly Room. It was apparently a pretty chaotic place. Bottles of fermenting bananas everywhere to feed the flies, thousands of little glass milk bottles each containing a different fly culture, the smell reportedly quite something. And they just bred flies, millions of them, looking for mutations, for unusual traits that popped up spontaneously. And in 1910, Morgan found one. A single male fly with white eyes instead of the normal red. That one white-eyed fly changed the course of biology. Morgan crossed it with normal red-eyed females, and the first generation all had red eyes, just like Mendel's tall peas. Red was dominant. But when he crossed those offspring with each other, the white-eyed trait reappeared in the next generation. And here's the crucial detail. It appeared almost exclusively in males. White-eyed females were extremely rare. The trait was somehow linked to sex. Now scientists already knew that sex in fruit flies, and in humans for that matter, was determined by chromosomes. Females have 2 X chromosomes, males have 1 X and 1 Y. And Morgan realized that the white-eye gene must be sitting on the X chromosome. That's why males showed it more often. They only have 1 X, so if that X carries the white-eye gene, there's no second X to mask it with a dominant red-eye gene. This was the first definitive proof that a specific gene was located on a specific chromosome. The chromosome theory of inheritance went from educated guess to established fact. Morgan's students went even further. Alfred Sturtevant, who was still an undergraduate at the time, think about that, an undergraduate, realized that by tracking how often different traits got inherited together, you could figure out how far apart the corresponding genes were on the chromosome. Genes that are close together on the same chromosome tend to be inherited together. Genes that are far apart get separated more often during the shuffling that happens when reproductive cells are made. Using this logic, Sturtevant stayed up one night in 1911 and created the first genetic map, a linear diagram showing the relative positions of genes along a chromosome. He was 19 years old. He later said it was probably the most important thing he ever did, and he did it in a single evening, putting off his homework to do it. So by the early 20th century, the picture was coming together beautifully. Hereditary information comes in discrete units called genes. Genes live on chromosomes. Chromosomes live in the nucleus of the cell. And chromosomes are made of what, exactly? What is the physical substance that carries genetic information? This is where things get chemically interesting. Scientists knew that chromosomes were made of 2 main types of molecules, proteins and a substance called nucleic acid. And for a long time, decades really, most biologists assumed that the important stuff, the actual carrier of genetic information, had to be the protein. and you can understand why they thought that. Proteins are spectacularly complex molecules. They come in an almost infinite variety of shapes and sizes. They're made of 20 different building blocks called amino acids, which can be strung together in any order, giving you an astronomical number of possible combinations. Nucleic acid, on the other hand, seemed boring by comparison. It was made of only 4 building blocks, 4 types of nucleotides. How could something with only 4 letters in its alphabet possibly encode the instructions for building something as complex as a human being? It seemed too simple, too monotonous. Surely the proteins were the smart molecules, and the nucleic acid was just structural scaffolding, something that held the chromosome together. This assumption turned out to be spectacularly wrong, and the story of how it was overturned is one of the great detective stories in science. It starts with a British medical officer named Frederick Griffith, working in London in the 1920s. Griffith was studying pneumonia, specifically the bacterium Streptococcus pneumonia, which was a major killer at the time. He was working with 2 strains, a virulent strain that caused disease and had a smooth outer coat, and a harmless strain that lacked the coat and looked rough under the microscope. Smooth and deadly, rough and harmless. Griffith did an experiment in 1928 that produced a result so bizarre it kept scientists puzzling for years. He took the deadly smooth bacteria and killed them with heat. Dead bacteria, completely dead, couldn't cause disease on their own. Then he mixed those dead smooth bacteria with living rough bacteria, the harmless ones, and he injected this mixture into mice. Now, you'd expect nothing bad to happen, right? The smooth ones are dead, the rough ones are harmless. But the mice died, and when Griffith examined their blood, he found living smooth bacteria. Somehow, the dead smooth bacteria had transformed the living rough bacteria into deadly smooth ones. Something had passed from the dead cells to the living cells, some transforming principle that carried the instructions for making the smooth coat. And whatever that substance was, it was capable of carrying hereditary information from one organism to another. But what was it? Griffith didn't know. He died in 1941 during the London Blitz, never having identified the molecule. But his experiment lit a fuse that burned for 16 years before someone finally answered the question. That someone was Oswald Avery, a quiet, meticulous researcher at the Rockefeller Institute in New York City. Avery was already in his 60s when he zeroed in on Griffith's transforming principle, and he'd spent most of his career studying pneumococcus, the same bacterium Griffith had worked with. Avery was not a flashy scientist. He was careful, methodical, almost painfully cautious, and he spent years, systematically stripping away components of the dead, smooth bacteria to figure out which molecule was doing the transforming. His approach was elegant in its logic. He and his colleagues, Colin MacLeod and Macklin McCarty, took extracts from the smooth bacteria and treated them with different enzymes. Enzymes that destroyed proteins, enzymes that destroyed lipids, enzymes that destroyed RNA. Each time, the transforming principle still worked. The rough bacteria still turned smooth. But when they added an enzyme that specifically destroyed DNA, deoxyribonuclease, the transformation stopped. Dead. No more smooth bacteria. The transforming principle was DNA. They published this result in 1944, and you might think the scientific world would have erupted. Here was clear evidence that DNA, not protein, carried genetic information, but the reaction was surprisingly muted. A lot of scientists simply didn't believe it. The protein camp was deeply entrenched. People argued that maybe Avery's DNA preparations were contaminated with tiny amounts of protein, and it was the protein doing the work. Others just couldn't wrap their heads around how a molecule with only 4 building blocks could encode complex instructions. Avery himself was cautious in his paper, carefully hedging his conclusions, which may have actually worked against him. He never won the Nobel Prize for this discovery, which is widely considered one of the greatest oversights in the history of the Nobel. But the evidence kept piling up. In 1952, Alfred Hershey and Martha Chase at Cold Spring Harbor Laboratory did an experiment that was almost brutally simple and incredibly clever. They worked with bacteriophages, viruses that infect bacteria. These phages are basically just a protein shell with DNA inside. When a phage infects a bacterium, it injects something into the cell, and that something hijacks the cell's machinery to make more phages. But what gets injected? The protein or the DNA? Hershey and Chase used radioactive labels to find out. They tagged the protein with radioactive sulfur, because proteins contain sulfur and DNA doesn't, and they tagged the DNA with radioactive phosphorus, because DNA contains phosphorus and proteins don't. Then they let the labeled phages infect bacteria, and they used a kitchen blender, literally a wearing blender, to shake the empty phage shells off the outside of the bacteria. When they checked where the radioactivity ended up, the answer was unambiguous. The radioactive phosphorus, the DNA label, was inside the bacteria, doing the work of infection. The radioactive sulfur, the protein label, was outside, in the discarded shells. DNA was the genetic material, not protein, DNA. And now the race was on, because once you know that DNA carries the instructions for life, the next question is obvious. What does it look like? What's its structure? And how does that structure allow it to do what it does? Store information, copy itself, pass instructions from parent to child? This is where we get to one of the most famous stories in all of science, and it's worth slowing down here because it's a story about ambition, competition, brilliance, and also some real injustice. By the early 1950s, several groups were trying to figure out the structure of DNA. The main players were in 2 cities, London and Cambridge. In London, at King's College, there was Maurice Wilkins and Rosalind Franklin. Franklin was a physical chemist and an expert in X-ray crystallography, a technique where you fire X-rays at a crystal of a substance, and the pattern of diffraction tells you about the arrangement of atoms inside. It's painstaking, demanding work, and Franklin was extraordinarily good at it. She produced the sharpest, clearest X-ray images of DNA that anyone had ever seen. In Cambridge, at the Cavendish Laboratory, there were James Watson and Francis Crick. Watson was an American biologist, young, brash, only 23 when he arrived in Cambridge. Crick was a British physicist who'd turned to biology, 12 years older than Watson, talkative, confident, brilliant at building theoretical models. The two of them didn't do experiments on DNA themselves. They built physical models, like Tinker Toys, trying to figure out how the atoms could fit together in a way that was consistent with the available data. Here's where the story gets complicated, and honestly, a little uncomfortable. Watson and Crick got access to Franklin's X-ray data without her. Explicit permission. Wilkins showed Watson one of Franklin's photographs, the famous Photo 51, and Watson immediately recognized its significance. The pattern of spots in that image told him that DNA had a helical structure. Franklin's own analysis, shared in a research report that was passed along through official but somewhat backdoor channels, contained crucial measurements: the spacing between repeating units, the dimensions of the helix. Watson and Crick used this data to constrain their model building, and in April 1953, they published their famous paper in Nature, Molecular Structure of Nucleic Acids, a structure for deoxyribose nucleic acid. It's barely more than a page long. One of the most important scientific papers ever written, and it's shorter than most emails you've sent. The structure they proposed was the double helix. And here's what's so beautiful about it. The structure immediately suggests the mechanism. Picture a twisted ladder. The sides of the ladder are made of alternating sugar and phosphate groups. That's the backbone, the structural part. The rungs of the ladder are pairs of those 4 nucleotide bases, adenine, thymine, guanine, and cytosine, A, T, G, and C. And here's the key insight. The bases pair up in a specific way. Adenine always pairs with thymine. Guanine always pairs with cytosine. Always. This is called complementary base pairing, and it's held together by hydrogen bonds, gentle, reversible connections, like 2 pieces of Velcro that you can pull apart and stick back together. Think about what that means. If you know the sequence of bases on one strand, you automatically know the sequence on the other strand. If one side reads ATGC, the other side must read TACG. The 2 strands are mirror images of each other, informationally speaking. And this immediately tells you how DNA can copy itself. You unzip the 2 strands, and each strand serves as a template for building a new partner. Every A gets a new T, every C gets a new G. You end up with 2 identical copies of the original molecule. Watson and Crick knew this. Their paper contains one of the most famous understatements in scientific history. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. That's British understatement at its finest. What they were really saying was, we've figured out how life copies itself. Now, the 4-letter alphabet that had seemed too simple, it turns out to be more than enough. Think of it like binary code in computers, just zeros and ones, 2 symbols, and yet you can encode anything. Music, images, entire operating systems. DNA uses 4 symbols instead of 2, and the information is encoded in the sequence, the order of the bases along the strand. A gene is essentially a stretch of DNA with a particular sequence of A's, T's, G's, and C's, and that sequence spells out instructions. The human genome contains about 3 billion base pairs, 3 billion rungs on that twisted ladder, packed into nearly every cell in your body. That's an enormous amount of information encoded in just 4 letters. Watson, Crick, and Wilkins shared the Nobel Prize in Physiology or Medicine in 1962. Rosalind Franklin did not. She had died of ovarian cancer in 1958 at the age of 37, possibly caused by her extensive exposure to X-rays during her research. The Nobel Prize is not awarded posthumously, so she was ineligible. But even setting that aside, there's a real question about whether she would have been included had she lived. Watson's later memoir, The Double Helix, portrayed her dismissively, even mockingly, and it took decades for the scientific community to fully recognize how essential her work had been. Her X-ray data wasn't just helpful, it was foundational. Without Photo 51, without her precise measurements, Watson and Crick's model might have taken years longer to build, if they'd built it at all. It's one of those stories that reminds you that science is done by human beings, with all the messiness that implies. But let's come back to the molecule itself, because knowing the structure is only the beginning. The next question is, How does a cell actually read the information stored in DNA? You've got this long string of A's, T's, G's, and C's, and somehow that has to turn into a living, functioning cell with thousands of different proteins, doing thousands of different jobs. The process of going from DNA to protein is so central to biology that Francis Crick called it the central dogma, a term he later admitted was probably too strong, but it stuck. The idea is this: DNA makes RNA, and RNA makes protein. Information flows in one direction, from the archive to the working copy to the final product. Let's unpack that, because it's one of those things that sounds simple when you state it, but the machinery involved is breathtaking. The first step is called transcription, and the name is apt. It's like transcribing a document, making a copy in a slightly different format. Inside the nucleus of your cell, an enzyme called RNA polymerase lands on a stretch of DNA and begins reading one strand. As it moves along, it builds a complementary strand of messenger RNA, mRNA. It's similar to DNA, but single-stranded, and it uses the base uracil instead of thymine. So where DNA has ATGC, the mRNA copy reads UACG. This mRNA molecule is basically a portable copy of the gene, a set of instructions that can leave the nucleus and travel out into the cell where the protein-building machinery lives. And that machinery is the ribosome. If DNA is the blueprint archive and mRNA is the work order, then the ribosome is the factory floor. Ribosomes are themselves made partly of RNA and partly of protein, and they're ancient. So ancient that they're nearly identical in bacteria and in human cells, which tells you they were perfected very early in the history of life and were so good at their job that evolution barely touched them for billions of years. When an mRNA molecule reaches a ribosome, the ribosome begins reading it 3 bases at a time. Each group of 3 bases, called a codon, specifies a particular amino acid. There are 64 possible codons, and they map to 20 different amino acids, plus a few stop signals. This mapping, this genetic code, is essentially universal across all life on Earth. The codon GUC means the amino acid valine, whether you're a bacterium, a mushroom, or a blue whale. That universality is one of the most profound facts in biology, because it means all life shares a common ancestor, a single origin point somewhere in the deep past. So the ribosome reads the codons one by one, and for each codon, a small molecule called transfer RNA, tRNA, brings the matching amino acid. The amino acids are linked together, one after another, into a growing chain, and that chain, that string of amino acids, is a protein. But here's the thing. A protein isn't just a string. As the chain grows, it begins to fold. The amino acids interact with each other, attracted here, repelled there, forming loops and sheets and helices, and within milliseconds, the chain collapses into a precise 3-dimensional shape. And that shape is everything. The shape determines what the protein does, whether it's an enzyme that speeds up a chemical reaction, a structural beam that gives the cell its shape, a channel that lets specific molecules pass through a membrane, or a signal that tells another cell what to do. A protein's function is its shape, and its shape is determined by its sequence, and its sequence is determined by the DNA. That's the chain. That's how information becomes action inside a living cell. And this brings us to something I've been wanting to talk about, because it's one of those structures that doesn't get nearly enough appreciation for how clever it is. The cell membrane. Every cell in your body is wrapped in a membrane that is only about 7 to 8 nanometers thick. That's roughly 10 millionths of a millimeter. If you scaled a cell up to the size of a gymnasium, the membrane would be about as thick as a sheet of plastic wrap. And yet, this impossibly thin barrier is what separates the inside of the cell from the outside. It's what makes a cell, a contained, organized, living thing, rather than just a puddle of molecules dissolving into the surrounding water. So how does something that thin actually work? The answer starts with a peculiar kind of molecule called a phospholipid. And the best way to picture a phospholipid is to think of it as a tiny lollipop with 2 sticks instead of 1. The round head of the lollipop is hydrophilic. It loves water. It's attracted to water molecules. It's perfectly happy being surrounded by them. But the 2 tails dangling below are hydrophobic. They hate water. They're made of long, fatty acid chains, and they want nothing to do with water molecules. They'd much rather be near other fatty, oily molecules. Now, here's what's clever. When you dump a bunch of these phospholipids into water, they spontaneously arrange themselves into a double layer, a bilayer. The hydrophilic heads face outward on both sides, happily touching the watery environment, and the hydrophobic tails face inward, tucked away from water, nestled against each other in the middle of the sandwich. No one has to build this structure. No enzyme has to assemble it. It just happens, driven purely by the physics of how water and oil interact. It self-assembles, and that bilayer is the foundation of every cell membrane in every living thing on this planet. Think about what that means for a second. The basic architecture of the boundary that defines life isn't some incredibly complex engineered structure. It's an emergent property of how certain molecules behave in water. It's almost embarrassingly simple in concept, and yet it's been the defining feature of cellular life for nearly 4 billion years. The earliest cells, those first protocells drifting in ancient oceans, were probably little more than self-assembled lipid bubbles with some rudimentary chemistry happening inside. The membrane came first, or at least very early, because without a boundary, you can't concentrate anything. You can't keep your useful molecules from drifting away. You can't maintain the chemical conditions needed for life's reactions. The membrane is the prerequisite. But here's the thing. If the membrane were just a simple lipid bilayer and nothing else, cells would be in trouble. Because a pure lipid bilayer is pretty good at keeping large molecules and charged particles out, but it's also pretty good at keeping everything out, including things the cell desperately needs: glucose, amino acids, ions like sodium and potassium and calcium. These are all molecules that can't easily pass through a wall of fatty acid tails on their own. They're either too big, too charged, or too polar. So the cell needs doors, it needs gates, it needs selective passageways that let the right stuff in and keep the wrong stuff out. And ideally, it wants to control when those doors open and close. And this is where the membrane goes from being a simple barrier to being one of the most sophisticated structures in all of biology. Embedded in that lipid bilayer are thousands of proteins, membrane proteins, and they do an astonishing variety of jobs. Some of them are channels, literal tunnels through the membrane that allow specific ions to flow through. A potassium channel, for instance, is so precisely shaped that it can distinguish between a potassium ion and a sodium ion, even though sodium is actually smaller. That's remarkable selectivity. The channel is tuned to recognize potassium's specific size and charge distribution, and it lets potassium through while rejecting sodium almost completely. Roderick McKinnon won the Nobel Prize in Chemistry in 2003 for figuring out the atomic structure of potassium channels. And when people saw the structure for the first time, the elegance of it was stunning. A narrow selectivity filter, just a few atoms wide, lined with precisely positioned oxygen atoms that mimic the water molecules that normally surround a potassium ion, essentially tricking the ion into letting go of its water coat and slipping through. Other membrane proteins are pumps. They don't just let things flow passively. They actively shove molecules from one side of the membrane to the other, even against the concentration gradient, even when the molecules would naturally want to flow the other direction. This takes energy, of course, and the most famous of these pumps is the sodium-potassium pump, which uses one molecule of ATP, the cell's energy currency, to push 3 sodium ions out of the cell and pull 2 potassium ions in. Every single one of your cells is running this pump constantly, millions of copies per cell, burning through a significant fraction of all the energy your body produces just to maintain the right balance of sodium and potassium across the membrane. Some estimates suggest that the sodium-potassium pump alone accounts for roughly 20 to 30% of your resting energy expenditure. You're burning calories right now, lying here in the dark, just to keep your ions sorted. And you need them sorted because that imbalance, more sodium outside, more potassium inside, creates an electrical voltage across the membrane, a resting membrane potential of about -70 millivolts. It's a tiny battery. Every cell is a tiny battery, and your nerve cells exploit that battery to send electrical signals. When a nerve fires, specific sodium channels snap open, sodium rushes in, the voltage flips, and that flip cascades down the length of the nerve cell like a wave, carrying information from your fingertip to your brain, or from your brain to your muscles. That's how you feel things. That's how you move. That's how you're processing these words right now, even as you're drifting toward sleep. So the membrane isn't just a wall. It's more like the control panel of the cell, studded with sensors and channels and pumps and receptors that let the cell interact with its environment in incredibly sophisticated ways. Receptor proteins on the cell surface can detect specific signaling molecules, hormones, neurotransmitters, growth factors, and when the right molecule binds to the right receptor, it triggers a cascade of events inside the cell. This is how your cells communicate with each other. When you eat a meal and your blood sugar rises, your pancreas releases insulin, and insulin molecules travel through your bloodstream until they encounter cells with insulin receptors on their surface. The insulin binds to the receptor changes shape, and that shape change sets off a chain reaction inside the cell that ultimately causes glucose transporters to move to the cell surface and start pulling glucose in. The whole thing is like a lock and key system, exquisitely specific, tightly regulated, and when it goes wrong, you get diseases like diabetes. And I want to linger on that idea of things going wrong, because the membrane is also where a lot of medicine happens. Many drugs work by interacting with membrane proteins. Beta blockers, which are used to treat high blood pressure and heart conditions, work by binding to specific receptors on heart muscle cells and blocking the effects of adrenaline. Antihistamines block histamine receptors. SSRIs, the most common antidepressants, work at the membrane of nerve cells by blocking the reuptake of serotonin. They interfere with a membrane transport protein that would normally suck serotonin back into the cell, so instead, the serotonin lingers in the gap between nerve cells and keeps signaling for longer. When you take a medication, there's a very good chance it's doing its work right there at the membrane, interacting with one of those thousands of specialized proteins embedded in that 7-nanometer thick lipid bilayer. Now, the membrane is also not static. It's not rigid like a wall. This is one of those things that changed how biologists thought about cells. In 1972, S. Jonathan Singer and Garth Nicholson proposed what they called the fluid mosaic model, and it's still the framework we use today. The idea is that the lipid bilayer behaves like a two-dimensional fluid. The phospholipids are constantly moving, sliding past each other laterally, swapping places, diffusing around the surface of the cell. And the proteins embedded in this fluid are like icebergs floating in a sea. They drift, they cluster, they rearrange. The whole membrane is dynamic, constantly shifting, reshaping itself. Cells can adjust the composition of their membranes, changing the types of lipids to make the membrane more fluid or more rigid depending on conditions. Bacteria that live in cold environments, for instance, incorporate more unsaturated fatty acids into their membranes to keep them fluid at low temperatures. The kinks in unsaturated fatty acid tails prevent them from packing together too tightly, kind of like how a pile of bent sticks won't stack as neatly as a pile of straight ones. This fluidity matters because cells need to do things with their membranes that a rigid wall could never do. Cells engulf particles by wrapping their membrane around them and pinching off a little bubble. That's called endocytosis, and it's how your immune cells eat bacteria. Cells secrete proteins by fusing little membrane-bound packages with the outer membrane and dumping the contents outside. That's exocytosis. Cells divide by pinching their membrane in half. None of this works if the membrane is stiff. It has to flow. And this brings us somewhere I've been wanting to go for a while now, because inside the cell, wrapped in their own membranes, are structures that have one of the most extraordinary origin stories in all of biology. I'm talking about mitochondria. You've probably heard mitochondria called the powerhouse of the cell, and that's accurate as far as it goes, but it barely scratches the surface of what makes them fascinating. Mitochondria are where your cells perform aerobic respiration, the process that takes the food you eat, combines it with the oxygen you breathe, and converts it into ATP, the molecule that powers almost everything your cells do. A typical human cell contains somewhere between a few hundred and several thousand mitochondria, depending on how energy-hungry that cell is. Muscle cells and heart cells are packed with them. Red blood cells, interestingly, have none. They jettison their mitochondria during development, which is a whole other story. But here's what's really wild about mitochondria. They have their own DNA. Not nuclear DNA, not the chromosomes we talked about earlier sitting in the nucleus. Mitochondria carry their own small circular genome, completely separate from the cell's main genetic material. And their DNA looks nothing like your nuclear DNA. It looks like bacterial DNA. Circular, compact, no introns, no histones wrapping it up. Mitochondria also divide on their own, splitting in two by a process that looks a lot like bacterial cell division. They have double membranes, an outer membrane, and a highly folded inner membrane. And that inner membrane is where the real energy-producing machinery lives. And in the late 1960s, a biologist named Lynn Margulis put all of these clues together and proposed something that most of her colleagues thought was absolutely wild. She proposed that mitochondria were once free-living bacteria, that billions of years ago, a larger cell engulfed a smaller, oxygen-breathing bacterium, and instead of digesting it, the two struck up a partnership. The smaller cell kept doing what it did best, converting oxygen and nutrients into energy, and the larger cell provided protection, raw materials, a stable environment. Over vast stretches of time, the two became so dependent on each other that neither could survive alone. The bacterium became the mitochondrion, and every mitochondrion in every one of your cells today is a descendant of that ancient partnership. Now, Margulis didn't come up with this idea entirely from scratch. The notion that organelles might have bacterial origins had been floated before, as far back as the early 1900s, by researchers like Konstantin Mereshkovsky and Ivan Wallin. But those early proposals were mostly speculative, and they were dismissed or ignored. What Margulis did was marshal the evidence: the double membranes, the circular DNA, the bacterial-style ribosomes inside mitochondria, the way they replicate independently, and weave it into a coherent, testable theory. She called it endosymbiosis, from the Greek for living together inside, and she laid it all out in a paper she submitted in 1966, and it was rejected. Not once, not twice. The paper was rejected by about 15 journals before it was finally published in 1967 in the Journal of Theoretical Biology. Her colleagues didn't just disagree with her. Many of them thought the idea was ridiculous. The prevailing view at the time was that organelles like mitochondria had evolved gradually from within the cell, that they were homegrown structures, not captured invaders. The idea that a fundamental part of your own cells was essentially a domesticated bacterium struck many biologists as too strange, too radical, maybe even a little absurd. But Margulis was persistent. She was, by all accounts, extraordinarily stubborn in the best possible way. The kind of scientist who doesn't flinch when the establishment pushes back, because she knows the evidence is on her side. And over the next 2 decades, the evidence kept piling up. When molecular biology techniques advanced enough to sequence mitochondrial DNA and compare it to bacterial genomes, the results were striking. Mitochondrial DNA is far more similar to the DNA of a group of bacteria called alpha proteobacteria than it is to the nuclear DNA of the cell it lives inside. That's a remarkable thing. The genome inside your own mitochondria is more closely related to a free-living soil bacterium than it is to the genome in your own nucleus. And the same story turned out to be true for chloroplasts, the organelles in plant cells that perform photosynthesis. They have their own DNA too, and it looks like it came from cyanobacteria, photosynthetic bacteria, that were engulfed in a separate endosymbiotic event. Margulis had proposed this as well, and she was right about both. By the 1980s and 1990s, endosymbiosis had gone from fringe hypothesis to textbook fact. It's now one of the most well-supported ideas in all of biology, and there's something deeply beautiful about it, if you think about it. You are not a single lineage of life. You are a collaboration. Every cell in your body is a partnership between at least 2 ancient organisms that found a way to work together so successfully that they became inseparable. Your cells are, in a very real sense, communities. And here's something else worth knowing about mitochondrial DNA, something that connects back to the story of inheritance we talked about earlier. Mitochondrial DNA is inherited almost exclusively from your mother. When a sperm cell fertilizes an egg, the sperm contributes its nuclear DNA, half your chromosomes, but its mitochondria are typically destroyed or diluted out. The egg cell provides essentially all the mitochondria for the resulting embryo. So your mitochondrial DNA is your mother's, and her mother's before her, and so on, stretching back through an unbroken maternal line. Geneticists have used this fact to trace human migration patterns, estimate when populations diverged, even identify what they call mitochondrial Eve, not a literal single woman, but a statistical construct representing the most recent common maternal ancestor of all living humans, estimated to have lived roughly 150,000 to 200,000 years ago in Africa. All of that from the DNA of an organelle that used to be a free-living bacterium. It's one of those facts that just sits with you. But anyway, let's come back to what mitochondria actually do inside your cells, because the energy production process is genuinely one of the most elegant pieces of molecular engineering you'll ever encounter. We said earlier that mitochondria perform aerobic respiration. They take glucose that's been partially broken down in the cytoplasm through a process called glycolysis, and they finish the job, extracting far more energy than glycolysis alone can provide. Glycolysis, which happens outside the mitochondria in the cell's cytoplasm, breaks one molecule of glucose into 2 molecules of pyruvate and nets you about 2 molecules of ATP. That's it. 2. And for the first couple billion years of life on Earth, that's roughly all cells could manage, because there wasn't much free oxygen in the atmosphere to work with. But once photosynthetic organisms, those ancient cyanobacteria, started pumping oxygen into the atmosphere, a new metabolic pathway became possible. Inside the mitochondria, pyruvate gets fed into a cycle called the citric acid cycle, or the Krebs cycle, named after Hans Krebs, the German-born British biochemist who worked out the details in the 1930s. Krebs himself had fled Nazi Germany in 1933. He was Jewish, and he lost his university position when the regime began purging Jewish academics. He ended up in England, at the University of Sheffield, working with limited resources, and it was there that he pieced together this elegant circular pathway. The Krebs cycle doesn't produce much ATP directly, but what it does is strip high-energy electrons from the breakdown products of glucose and hand them off to carrier molecules. Think of these carriers as little shuttle buses loaded with energetic passengers, and they're heading for the main event. That main event is the electron transport chain, and it takes place on the inner membrane of the mitochondria. That highly folded inner membrane I mentioned, with all those folds called cristae that massively increase the surface area. The electron transport chain is a series of protein complexes embedded in that membrane, and as electrons get passed from one complex to the next, energy is released at each step. That energy is used to pump hydrogen ions, protons, across the inner membrane, from one side to the other, building up a concentration gradient. You end up with a lot more protons on one side of the membrane than the other, and that imbalance represents stored energy, like water behind a dam. And then comes the part that won a Nobel Prize. The protons flow back through a remarkable molecular machine called ATP synthase, and as they flow through it, this machine literally spins. It physically rotates like a tiny turbine, and that rotation drives the chemical reaction that attaches a phosphate group to ADP, creating ATP. Peter Mitchell proposed the basic idea of this proton gradient driving ATP synthesis in 1961 in what he called the chemiosmotic hypothesis, and like Margulis, he faced years of skepticism before being vindicated. He won the Nobel Prize in Chemistry in 1978. And then in 1997, Paul Boyer and John Walker shared the Nobel Prize for working out the actual structure and mechanism of ATP synthase, revealing that it really does work like a rotary motor. The whole thing is maybe 10 nanometers across, and it spins at about 100 revolutions per second, cranking out ATP molecules as it goes. The total yield? From one molecule of glucose, the complete process, glycolysis plus the Krebs cycle plus the electron transport chain, produces somewhere around 30 to 36 molecules of ATP, depending on conditions. Compare that to the measly 2 from glycolysis alone. That's roughly a 15-fold increase in energy extraction, and it's why aerobic life was such a game changer. It's why you can run and think and maintain a body temperature of 37 degrees Celsius and do all the millions of energy-demanding things your cells do every second. your body produces and recycles roughly your own body weight in ATP every single day. Think about that. If you weigh 70 kilograms, your cells are manufacturing and consuming something like 70 kilograms of ATP in a 24-hour period. You don't stockpile it. You make it and use it almost instantly, in a constant, furious cycle. And when mitochondria malfunction, the consequences can be devastating. Because mitochondrial DNA is small and lacks many of the repair mechanisms that protect nuclear DNA, it's particularly vulnerable to mutations. Mitochondrial diseases, conditions caused by defects in mitochondrial DNA or in the nuclear genes that code for mitochondrial proteins, can affect almost any organ system, but they tend to hit the most energy-hungry tissues hardest: the brain, the heart, the muscles, the liver. Symptoms can range from mild exercise intolerance to severe neurological degeneration, organ failure, and death in childhood. These diseases are rare individually, but collectively they affect roughly 1 in every 5,000 people, and they remain among the most difficult conditions in medicine to treat because you can't easily fix or replace the DNA inside thousands of mitochondria scattered through billions of cells. There's actually been a remarkable and controversial medical advance related to this. In 2015, the United Kingdom became the first country to legalize a technique called mitochondrial replacement therapy, sometimes called 3-parent IVF in the press, though that's a bit misleading. The idea is that if a mother carries a mitochondrial disease mutation, doctors can take the nuclear DNA from her egg and transfer it into a donor egg that has healthy mitochondria, effectively giving the resulting child nuclear DNA from both parents, but mitochondrial DNA from a third person, the donor. The child ends up with genetic material from 3 people, which is what generated the headlines. But the mitochondrial DNA contribution is tiny, only 37 genes, compared to the roughly 20,000 protein-coding genes in the nuclear genome. Still, it raised profound ethical questions about genetic modification of the human germline, because those donated mitochondria would be passed down to future generations through the maternal line. So mitochondria sit at this incredible intersection of evolutionary history, energy metabolism, genetics, medicine, and ethics. They're not just powerhouses. They're ancient symbionts with their own genome, their own replication machinery, their own vulnerabilities, and they remind us that the cell is not a single, unified thing. It's a coalition, an assembly of parts with different origins that have learned to work together so seamlessly that we forget they were ever separate. Now, all of this energy that mitochondria produce, all that ATP, it has to go somewhere, right? It has to power something. And one of the most important things it powers, one of the most fundamental processes in all of biology, is cell division. Because your body doesn't just maintain itself, it grows, it heals, it replaces worn-out cells, and to do any of that, cells have to copy themselves. And copying a cell is not a simple task. You have to duplicate 3 billion base pairs of DNA with almost perfect accuracy. You have to organize those duplicated chromosomes so each daughter cell gets exactly the right set. You have to divide the cytoplasm, the organelles, the membranes, and you have to do it all in a coordinated sequence where each step depends on the one before it. The process that handles this is called mitosis, and it's one of the most tightly regulated operations in all of cellular biology. And when that regulation breaks down, when the controls fail and cells start dividing when they shouldn't, or refuse to stop when they should, That's when you get cancer. And cancer, at its core, is not a disease of foreign invaders or mysterious poisons. It's a disease of the cell's own machinery. It's what happens when the instruction manual for division gets corrupted, when the brakes fail, when the quality control systems that normally catch mistakes and say, "Stop, something's wrong here," go silent. To really understand that, though, you need to understand what normal cell division looks like when everything goes right. Because honestly, when everything goes right, it's breathtaking. So let's walk through it. Mitosis. Your cells do this millions of times a day, and most of the time you never notice. Right now, deep in your bone marrow, cells are dividing to make fresh blood cells. In your gut lining, cells are dividing to replace the ones that get scraped away by the simple act of digesting food. In your skin, cells are dividing to patch up that little cut you got 2 days ago that you've already forgotten about. Each one of those divisions follows the same ancient choreography, and it starts long before the cell actually splits in two. The first thing that has to happen is that the cell needs to copy all of its DNA. Every single chromosome, all 46 of them, all 3 billion base pairs, all of it. This happens during a phase called S phase, S for synthesis, and it takes several hours. The cell unzips its double helix and builds a complementary copy of each strand, using the same base pairing rules Watson and Crick described. A pairs with T, C pairs with G. The enzyme doing the heavy lifting is called DNA polymerase, and it's remarkably fast and remarkably accurate. It adds about 1,000 nucleotides per second in human cells and makes roughly 1 uncorrected error per billion base pairs. Think about that. If you were typing out 3 billion characters and you made 1 typo in the entire document, that's the kind of accuracy we're talking about. And even when it does make a mistake, there are proofreading enzymes that come along behind it and fix most of the errors. It's like having an editor who reads every word right after you type it. By the end of S phase, the cell has twice as much DNA as it started with. Each chromosome now consists of 2 identical copies, called sister chromatids, joined together at a pinch point called the centromere. The cell is ready. But it doesn't just immediately divide. There's a gap phase, G2, where the cell checks its work. Did the DNA copy correctly? Is there any damage? Are there enough raw materials and energy to complete division? This is one of several checkpoints built into the cell cycle, and they're crucial. If something's wrong, the cell pauses here. It tries to fix the problem. And if it can't fix it, in many cases, the cell will actually destroy itself rather than pass on damaged DNA. That self-destruct program is called apoptosis, programmed cell death, and it's one of the most important safety mechanisms in your body. We'll come back to that, but assuming everything checks out, the cell enters mitosis proper. And mitosis itself has these beautifully distinct stages that scientists identified over a century ago, back in the 1870s and 1880s, when a German biologist named Walter Flemming spent years staining cells with aniline dyes and watching them under the microscope. Flemming was the one who coined the term mitosis from the Greek word for thread because of how the chromosomes looked. These thread-like structures that appeared, moved, and then vanished again. He published his masterwork, a book called Zellsubstanz, Kern- und Zellteilung, in 1882, and his drawings of dividing cells are still remarkably accurate. He didn't know what chromosomes were made of. He didn't know about DNA. But he could see the dance, and he documented it with extraordinary care. Here's how the dance goes. In the first stage, prophase, those duplicated chromosomes, which have been loosely spread out through the nucleus as long, thin strands, begin to condense. They coil up tighter and tighter, like winding a garden hose into a compact loop, until they become short, thick, visible structures. At the same time, outside the nucleus, a structure called the mitotic spindle starts to form. It's made of protein filaments called microtubules, and it grows outward from two organizing centers that migrate to opposite poles of the cell, like setting up goalposts at either end of a field. Then the nuclear envelope, that membrane surrounding the nucleus, breaks apart. It just dissolves. And now the chromosomes are floating free in the cytoplasm, and the spindle fibers reach in and grab them. Each chromosome gets attached to spindle fibers from both poles, one set of fibers pulling toward one side, the other set pulling toward the other. And here's where there's another checkpoint, a really critical one. The cell actually pauses and checks. Is every single chromosome properly attached to spindle fibers from both sides? Because if even one chromosome is attached wrong, connected to fibers from only one pole, or not connected at all, the division has to stop. A protein complex called the spindle assembly checkpoint enforces this. It's like a foreman on a construction site who won't let the crane lift until every cable is properly secured. Once every chromosome passes inspection, the cell enters anaphase, and this is the moment that still gives me chills when I watch it in time-lapse microscopy. The sister chromatids, those identical copies that have been joined at the centromere, are pulled apart. The centromere splits, and the spindle fibers reel the separated chromosomes toward opposite poles of the cell. It happens with this smooth, deliberate precision. 46 chromosomes glide to one side, 46 to the other, and then the cell pinches in the middle. A contractile ring of protein filaments tightens like a drawstring, and you get two daughter cells, each with a complete identical set of chromosomes. The nuclear envelope reforms around each set, the chromosomes decondense back into their loose working form, and each new cell enters G1, the first gap phase, ready to go about its business. The whole process, from the start of mitosis to the end of cytokinesis, takes maybe an hour or two in most human cells. But the full cell cycle, including the DNA replication and the gap phases and the checkpoints, can take anywhere from about 24 hours in rapidly dividing cells to days, weeks, or even longer in slower tissues. Some cells, like neurons in your brain and muscle cells in your heart, essentially exit the cell cycle altogether. They enter a resting state called G0 and just stay there, doing their job, not dividing, sometimes for your entire life, which is why damage to those cells is so serious. They're not easily replaced. Now, all of those checkpoints I mentioned, all of that quality control, it exists for a reason. Because the consequences of getting division wrong are severe. If a cell divides with damaged DNA, or with the wrong number of chromosomes, the results can range from a dead cell to a malfunctioning cell to, in the worst case, a cell that has gained the ability to divide without restraint. And that's cancer. Here's the thing about cancer that I think surprises people when they first learn it. Cancer isn't one disease. It's hundreds of diseases, maybe thousands, all sharing one common feature: uncontrolled cell division. A breast cancer and a leukemia and a melanoma are very different diseases at the molecular level, caused by different mutations in different genes, behaving in different ways. But they all involve cells that have escaped the normal controls on growth. The genes that matter most fall into two broad categories. The first are called proto-oncogenes. In their normal form, these genes produce proteins that promote cell growth and division. They're the accelerator pedal. When a mutation turns a proto-oncogene into an oncogene, it's like the accelerator getting stuck to the floor. The cell gets a constant grow, divide signal even when it shouldn't. One of the most famous examples is a gene called RAS. About 30% of all human cancers involve a mutated RAS gene. 30%. It was first identified through studies of cancer-causing viruses in the 1960s and 1970s, and when researchers finally found mutant RAS in human tumor cells in the early 1980s, teams led by Robert Weinberg at MIT, Michael Wigler at Cold Spring Harbor, and Mariano Barbacid at the National Cancer Institute, all publishing within months of each other in 1982. It was a landmark moment. It showed that cancer could be driven by specific, identifiable mutations in specific genes. The second category is tumor suppressors. These are the brakes. They produce proteins that slow down division, trigger DNA repair, or activate apoptosis, that self-destruct program when something goes wrong. The most famous tumor suppressor is a gene called TP53, which makes a protein called p53. It's been called the guardian of the genome, and for good reason. When DNA gets damaged, p53 is the protein that stops the cell cycle, calls in repair crews, and if the damage is too severe, orders the cell to kill itself. In more than half of all human cancers, p53 is mutated or missing. The cell loses its most important safety inspector. The story of how p53 was discovered is itself a winding tale. It was first identified in 1979, independently by several groups, including David Lane in Dundee, Scotland, and Arnold Levine at Princeton. But initially, everyone thought it was an oncogene, a gas pedal, because they were finding it in tumor cells at high levels. It took nearly a decade to realize that the p53 they were seeing in tumors was a mutant form. The normal, wild-type p53 was actually a brake. The mutations weren't activating it. They were breaking it, and sometimes the broken version actively interfered with any remaining normal p53. It was like finding a broken brake pedal and initially assuming brakes must cause crashes. So cancer typically requires multiple hits. One mutation isn't usually enough. You need the accelerator stuck and the brakes broken, and often more than that. Mutations that help the tumor grow its own blood supply, a process called angiogenesis, and mutations that let cancer cells detach from their neighbors and spread to distant organs, which is metastasis. And that's what makes cancer truly lethal in most cases. This multi-hit model was first proposed by Alfred Knudsen in 1971, based on his studies of retinoblastoma, a childhood eye cancer. He noticed that kids who inherited one defective copy of a tumor suppressor gene, the RB gene, developed cancer much earlier and in both eyes, because they only needed one more mutation to knock out the remaining good copy. Kids without the inherited mutation needed two separate hits in the same cell, which was much rarer. Knudsen's two-hit hypothesis was elegant and powerful, and it's been validated and expanded many times since, and this is where the story connects back to everything we've been talking about: DNA replication fidelity, checkpoint proteins, repair enzymes, apoptosis. Every single one of those systems is a layer of defense against cancer. DNA polymerase's proofreading ability, the mismatch repair enzymes that catch what polymerase misses, the checkpoint proteins that halt the cell cycle when damage is detected, p53 standing guard, the spindle assembly checkpoint, making sure chromosomes segregate properly. All of it. Cancer is what happens when enough of these layers fail, in the same cell, at the same time. And here's something that puts it in perspective. Your body contains roughly 37 trillion cells. Billions of them divide every day. Each division copies 3 billion base pairs of DNA. The fact that cancer doesn't happen more often is, honestly, a testament to how extraordinarily good these systems are. The error rate is vanishingly small, but over a lifetime, across trillions and trillions of divisions, the odds eventually catch up with most of us. That's why cancer risk increases so dramatically with age. It's not that old cells are weaker exactly. It's that they've had more time to accumulate mutations, more rolls of the dice, more chances for that unlucky combination of hits. But cells don't just divide and sometimes go wrong. They also communicate. They talk to each other constantly, coordinating their behavior, deciding when to grow and when to stop, when to specialize, and when to die. And the way they do this, the signaling networks that connect cells into tissues, and tissues into organs, and organs into a functioning body, that's a whole other layer of complexity that makes the cell cycle look almost simple by comparison. Because the signals that tell a cell to divide don't originate inside the cell. They come from outside, from neighboring cells, from distant hormones carried in the blood, from the extracellular matrix, the scaffolding that holds tissues together. And the way those signals are received, interpreted, and acted upon involves some of the most intricate molecular machinery you'll find anywhere in biology. So let's talk about that for a second. Imagine you're a cell sitting inside a tissue. Say, a cell lining the wall of your small intestine. You're surrounded by other cells, packed in tight, and you're getting chemical messages from every direction. Some of those messages are telling you to stay put. Some are telling you to divide. Some are telling you to differentiate, to become more specialized. And a few might be telling you to die because you've been around long enough and it's time to make room for a fresh replacement. You can't just respond to all of these signals at once. That would be chaos. You have to receive each one, figure out what it means, weigh it against the other signals you're getting, and then make a decision. And the system that lets you do this is called signal transduction. Here's the basic idea. A signaling molecule, let's call it a ligand, floats up to the outside of the cell and binds to a receptor protein embedded in the cell membrane. We talked about membrane proteins before, how they sit in that phospholipid bilayer like icebergs, with parts sticking out on both sides. Receptor proteins are a specific type. Their outer portion is shaped to grab onto one particular ligand, like a lock fitting one key. When that ligand binds, the receptor changes shape, and here's the clever part. The shape change on the outside of the membrane causes a shape change on the inside. The signal has been transduced. It's been converted from an external chemical event into an internal mechanical one, without anything actually crossing the membrane. Now, that internal shape change triggers a cascade. Not just one reaction, but a whole chain of them, like dominoes falling. Each step in the chain typically involves one protein activating another protein, which activates another, which activates another. And at each step, the signal can be amplified. One receptor might activate 10 copies of the next protein. Each of those 10 might activate 10 more. By the time the signal reaches the nucleus, where it might switch on a gene, a single molecule binding to the cell surface could have triggered thousands or millions of downstream events. That amplification is crucial. It means cells can respond to incredibly faint signals, just a few molecules of a hormone in the bloodstream, and still mount a robust, decisive response. One of the most important signaling cascades in your body is the MAP kinase pathway, sometimes called the RAS-M, APK pathway. And yes, that's the same RAS we talked about with cancer. Here's how it works in a normal, healthy cell. A growth factor, say epidermal growth factor, EGF, binds to its receptor on the cell surface. The receptor activates RAS, that small G protein sitting on the inner face of the membrane. RAS activates a kinase called RAF. RAF activates MEK. MEK activates ERK, and ERK travels into the nucleus and turns on genes that push the cell toward division. Each step is a kinase, an enzyme that activates the next protein in line by sticking a phosphate group onto it, like flipping a switch. And each step amplifies the signal further. Think about what that means. The cell isn't just passively receiving orders. It's processing information. It's running the signal through multiple checkpoints, giving itself opportunities at every step to modulate the response, to dial it up or dial it down, to integrate it with other signals coming in through other pathways. And this is where it gets really beautiful. Because cells don't just have one signaling pathway. They have dozens, and those pathways crosstalk. The output of one pathway can influence another. A signal coming in through one receptor can be modified by a signal coming in through a completely different receptor. The cell is essentially running a computation, weighing inputs, integrating information, and arriving at a decision. This is why cancer is so hard to treat, by the way. When we talked about RAS being mutated in 30% of cancers, what's actually happening is that the RAS protein is stuck in its on position. It's sending a constant growth signal down the MAP kinase pathway, regardless of whether any growth factor is actually binding to the receptor. The cell thinks it's being told to divide all the time, when really it's just a broken switch. And you might think, okay, just block RAS. But RAS was considered undruggable for decades. Its surface is smooth and small, with no obvious pocket where a drug molecule could wedge in and shut it off. It wasn't until 2021 that the FDA approved sotorasib, the first drug to directly target a specific RAS mutation, KRAS G12C, found in some lung cancers. That took nearly 40 years from the discovery of oncogenic RAS to a drug that could actually hit it. 40 years of some of the smartest biochemists in the world working on a single protein. But anyway, let's zoom out from cancer for a moment, because signal transduction isn't just about growth. It's about everything. Your nervous system, for instance. When a nerve impulse reaches the end of a neuron, it triggers the release of neurotransmitters into the tiny gap between that neuron and the next one. Those neurotransmitters are ligands. They bind to receptors on the next neuron and trigger a response. That's signal transduction. When you smell coffee in the morning, odor molecules are binding to receptor proteins in your nasal epithelium, triggering cascades that eventually produce an electrical signal your brain interprets as that rich, warm, familiar smell. When your pancreas detects rising blood sugar after a meal and releases insulin, and that insulin travels through your bloodstream and binds to insulin receptors on muscle and fat cells, telling them to absorb glucose. That entire process is signal transduction, start to finish. And here's something worth sitting with. The insulin signaling pathway was one of the first to be really thoroughly mapped out, and the story behind it connects to one of the most important medical breakthroughs of the 20th century. Before 1921, a diagnosis of type 1 diabetes was essentially a death sentence. Your immune system destroys the beta cells in your pancreas that produce insulin, and without insulin, your cells can't take up glucose. You starve in the midst of plenty, glucose flooding your blood but unable to get inside the cells that need it. Frederick Banting and Charles Best, working in a borrowed lab at the University of Toronto with grueling, unglamorous experiments on dogs, managed to extract insulin from pancreatic tissue and inject it into diabetic patients. The results were dramatic. Children who had been wasting away, days from death, got up and walked. It was one of those moments in medical history that genuinely felt like a miracle. Banting received the Nobel Prize in 1923, just 2 years after the discovery, one of the fastest Nobel recognitions ever. But here's the thing. Banting and Best knew insulin worked. They didn't fully understand how. The receptor for insulin wasn't identified until the 1970s and 1980s through work by Pedro Cuatrecasas and others. The downstream signaling cascade, how insulin binding to its receptor actually causes glucose transporters to move to the cell surface, took even longer to unravel. It involves a pathway called PI3K-AKT, which, like the MAP kinase pathway, is a chain of kinases activating kinases, and this pathway too turns out to be deeply involved in cancer. The PI3K pathway is one of the most frequently mutated in human tumors. So here you have a signaling system that, when it works properly, lets your cells absorb sugar, and when it goes wrong, can contribute to uncontrolled growth. The same molecular machinery, repurposed by mutation into something dangerous. This is a pattern you see over and over in cell biology. The systems that keep you alive, the ones that coordinate growth, metabolism, survival, are the exact same systems that, when broken, cause disease. It's not that cancer invents new machinery. It hijacks existing machinery. It takes the accelerators and jams them on, or it takes the brakes and cuts the lines. And this insight, which really crystallized in the 1980s and 1990s as these pathways were mapped, fundamentally changed how we think about treating disease. Instead of just poisoning rapidly dividing cells with chemotherapy, which works, but damages healthy tissue too, you could try to design drugs that target specific broken components in specific pathways. That's the whole idea behind targeted therapy, and it's why understanding signal transduction at the molecular level isn't just academic. It's the foundation of modern precision medicine. Now, there's another kind of cell communication we should talk about, because it operates on a completely different scale, and it's one of the most profound questions in all of biology. How does a single fertilized egg, one cell with one genome, give rise to the roughly 200 different cell types in your body? Your neurons and your liver cells and your red blood cells all contain the same DNA. Every single one of them has the full set of instructions. So how does a neuron know to be a neuron? How does a muscle cell know to be a muscle cell? The answer lies in gene regulation, which genes are turned on and which are turned off in any given cell. And that regulation is controlled in large part by signals that cells send to each other during development. This brings us to one of the most elegant experiments in the history of biology, and to a scientist whose work reshaped our understanding of how bodies are built. In the 1920s, a German embryologist named Hans Spemann, working with his student Hilde Mangold, was studying newt embryos. They took a small piece of tissue from one specific region of an early embryo, a region called the dorsal lip of the blastopore, and transplanted it into a different embryo. What happened next was extraordinary. The transplanted tissue didn't just develop on its own. It recruited the cells around it, the host cells, and organized them into a second complete body axis, a second head, a second spinal cord, a second set of structures growing right alongside the original ones. The host embryo developed what was essentially a conjoined twin, and the remarkable thing was that most of the cells forming that second body plan belonged to the host. The transplanted tissue had somehow instructed its neighbors to change their fates, to become things they were never originally going to become. Spemann called this piece of tissue the organizer, and the name stuck because that's exactly what it did. It organized the cells around it into a coherent body plan. Hilda Mangold, tragically, never saw the full impact of this discovery. She died in a household accident in 1924, at just 26 years old, before the work was even published. Spemann went on to receive the Nobel Prize in 1935 for the discovery, and Mangold's contribution, while acknowledged in the original paper, it was her doctoral thesis, has only been fully appreciated by history in more recent decades. But the experiment itself opened up an entirely new way of thinking about development. It told biologists that cells don't just follow their own internal program in isolation. They listen to their neighbors. They receive signals, and those signals can completely rewrite what a cell becomes. Now, for decades after that experiment, the big mystery was, what is the signal? What molecule is the organizer actually secreting that tells surrounding cells to become neural tissue instead of skin? It took until the 1990s, 70 years later, before the molecular identity of the organizer's signals started to become clear, and the answer turned out to be wonderfully counterintuitive. The organizer doesn't work by sending an activating signal. It works by blocking an inhibitor. There's a family of signaling molecules called bone morphogenetic proteins, or BMPs, that are present throughout the early embryo, and by default, BMPs push cells toward becoming skin, toward becoming epidermis. The organizer secretes molecules, noggin, cordon, follistatin, that bind to BMPs and neutralize them. So the cells near the organizer are shielded from BMP signaling, and in the absence of that signal, their default fate shifts. They become neural tissue instead. It's like the organizer is creating a quiet zone, a shadow where the noise of BMP signaling can't reach, and in that silence, the nervous system begins to form. Think about how elegant that is. You don't need a special become a brain signal. You just need to block the become skin signal in the right place at the right time. And this principle, that development often works by inhibiting inhibitors, by removing constraints rather than adding instructions, turns out to be a recurring theme throughout embryology. This is where we get into the broader concept of morphogen gradients, and it's one of the most beautiful ideas in all of biology. A morphogen is a signaling molecule that spreads out from a source and forms a concentration gradient, high near the source, lower as you move away. And cells can read their position in that gradient and respond differently depending on how much morphogen they're exposed to. So a high concentration might trigger one set of genes, a medium concentration triggers a different set, and a low concentration triggers yet another. One signal, multiple outcomes, depending on dose. The idea was first proposed in a rigorous mathematical framework by Alan Turing. Yes, the same Alan Turing, the codebreaker, the father of computer science, in a remarkable 1952 paper called The Chemical Basis of Morphogenesis. Turing showed mathematically that simple reaction diffusion systems, where chemicals are produced, diffuse through tissue, and interact with each other, could spontaneously generate patterns, stripes, spots, waves, from initially uniform conditions. He was trying to explain how a symmetrical embryo breaks symmetry, how pattern emerges from uniformity. And while the specific mechanisms he proposed were theoretical, the core insight was prophetic. Biology confirmed it over and over in the decades that followed. One of the most famous examples involves a signaling molecule called sonic hedgehog, and yes, It's really called that, named after the video game character, because the gene was discovered in fruit flies in the 1980s, during a period when fly geneticists were giving genes wonderfully irreverent names. In the developing vertebrate embryo, sonic hedgehog is secreted from a structure called the notochord, and from a region at the base of the developing neural tube called the floor plate. It diffuses outward, forming a gradient, and different concentrations of sonic hedgehog specify different types of neurons at different positions in the spinal cord. The motor neurons that will eventually control your muscles form at one concentration. Different classes of interneurons form at other concentrations. The gradient literally tells cells where they are, and that positional information determines what they become. And here's what connects all of this back to everything we've been talking about for a while now. These morphogen signals, sonic hedgehog, BMPs, WNTS, FGFs, all these signaling families, they work through signal transduction pathways. The same kinds of receptor-ligand interactions, the same cascading phosphorylation events, the same transcription factor activation that we talked about earlier. The difference is that during development, the output isn't just divide or take up glucose. The output is turn on this specific combination of genes that will make you a motor neuron for the rest of your life. The signal is transient. It might last hours or days during a critical window of development. But the consequences are permanent, because the genes that get activated change the cell's identity in ways that are self-reinforcing. This is where we need to talk about transcription factors and gene regulation a bit more deeply, because this is really the heart of how one genome produces 200 cell types. Every cell in your body has the same DNA, roughly 20,000 protein-coding genes, plus all the regulatory sequences packed into those 46 chromosomes. But a liver cell might only express maybe 4,000 or 5,000 of those genes at any given time, and a neuron expresses a different but overlapping set. The difference between a liver cell and a neuron isn't what DNA they have. It's which parts of that DNA are accessible and actively being read. The control happens at multiple levels. There are transcription factors, proteins that bind to specific DNA sequences near genes and either promote or block transcription. A single transcription factor can regulate hundreds of target genes, and transcription factors often work in combinations, so the specific mix of transcription factors present in a cell determines which genes are on during development. Morphogen signals activate specific transcription factors, and those transcription factors activate other transcription factors, creating cascading regulatory networks that progressively narrow a cell's identity. An early embryonic cell has many possible fates. With each round of signaling and gene regulation, options are eliminated, commitments are made, until the cell arrives at its final differentiated state. But there's an even deeper layer to this, and it's called epigenetics. Beyond the transcription factors that are present right now, cells also carry a kind of molecular memory of which genes should be on and which should be off. This memory is encoded in chemical modifications to the DNA itself and to the histone proteins that DNA wraps around. Methyl groups can be added to cytosine bases in DNA, typically silencing nearby genes. Acetyl groups, methyl groups, and other chemical tags can be added to histone tails, either loosening or tightening the chromatin structure, making genes more or less accessible. These epigenetic marks are heritable. When a cell divides, the daughter cells inherit not just the DNA sequence, but also much of the epigenetic landscape, which is why a liver cell divides to produce more liver cells, not neurons. The identity is locked in, maintained across generations of cell division, even though the underlying DNA sequence is identical in every cell. This whole system, morphogen gradients establishing initial patterns, signal transduction pathways interpreting those gradients, transcription factor networks narrowing cell fate, epigenetic modifications locking in identity, it all works together to build a body from a single cell, and it happens with astonishing reliability. Every human embryo goes through essentially the same sequence of events. The fertilized egg divides, forms a ball of cells, then a hollow sphere, then gastrulates, folds in on itself to establish the three primary germ layers, ectoderm, mesoderm, and endoderm, which will give rise to skin and nerves, muscles and bones, gut and organs, respectively. Organizers and morphogen gradients pattern the body axes, head to tail, back to belly, left to right. Organs begin to form in the right places, limbs bud out, fingers separate, and all of this is orchestrated by cells talking to each other, sending and receiving signals, making decisions based on their position and their history. When it goes wrong, the consequences can be devastating: birth defects, developmental disorders, miscarriage. But what's honestly more astonishing is how rarely it goes catastrophically wrong, given the staggering complexity of the process. Billions of cells, coordinating across weeks and months, building structures of incredible intricacy, all from a set of instructions encoded in a molecule that's just 4 letters long. And that brings us gently to where all of this stands now, because the story of cell biology isn't finished. It's arguably entering its most remarkable phase. The tools available today would have seemed like science fiction to Hooke, peering at his cork, or to Leeuwenhoek, marveling at his animalcules. CRISPR-Cas9, the gene editing system discovered through studies of bacterial immune systems and developed into a precision tool by Jennifer Doudna and Emmanuelle Charpentier, who shared the Nobel Prize in 2020, lets researchers cut and edit DNA at specific locations with unprecedented accuracy. You can knock out a gene to see what it does. You can correct a mutation that causes disease. You can, in principle, rewrite the instructions. Single-cell RNA sequencing lets scientists read out which genes are active in individual cells, one cell at a time, across entire tissues. This has revealed cell types we didn't even know existed, subtle variations within what we thought were uniform populations. The Human Cell Atlas Project, launched in 2016, aims to map every cell type in the human body. Not just the traditional 200 or so categories, but potentially thousands of distinct states, each defined by its unique pattern of gene expression. Organoids, tiny, self-organizing structures grown from stem cells in a dish, can recapitulate aspects of organ development. Researchers have grown miniature brains, guts, kidneys, and lungs, not full organs, but simplified versions that capture enough of the real architecture to study disease and test drugs in ways that were never possible before. And all of it rests on the foundations we've been walking through together. The cell as the basic unit of life, the membrane that defines inside from outside, the mitochondria that power everything, the DNA that carries the instructions, the ribosomes that read them, the signal transduction pathways that let cells listen and respond, the checkpoints that keep division under control, the morphogen gradients that build pattern from uniformity. Every layer we've talked about connects to every other layer, because that's how biology works, not as isolated systems, but as an integrated, dynamic, endlessly communicating whole. You know, there's something deeply calming about that, if you sit with it for a moment. Right now, As you're lying here, your cells are doing all of this. Your mitochondria are spinning out ATP, billions of molecules every second, keeping everything running. Your membranes are maintaining their careful balance of ions and signals. Somewhere in your bone marrow, stem cells are dividing, producing fresh blood cells to replace the ones that wore out today. In your gut lining, cells that are only a few days old are carrying out their brief lives, absorbing nutrients before being shed and replaced. Your immune cells are patrolling, checking surfaces, reading molecular signatures, distinguishing self from non-self with extraordinary precision. Your neurons are firing in patterns that somehow add up to the experience of hearing these words, of feeling sleepy, of being you. None of it requires your conscious attention. None of it asks for your permission. It just happens, the way it's been happening in living things for nearly 4 billion years, since the first protocells stumbled into existence in some warm, shallow chemistry experiment on the early Earth. The machinery has been refined, elaborated, made more complex, more capable, more resilient. But the core logic, the basic cellular operating system, is ancient. Your cells speak a molecular language that was old before the first animals appeared, before the first plants colonized land, before the first eukaryote swallowed that fateful bacterium and gained its mitochondria. And that's maybe the most wonderful thing about all of this. You are made of cells, and those cells are made of molecules, and those molecules follow the laws of chemistry and physics. And yet somehow, out of all that mechanism, all those cascades and gradients and feedback loops, here you are, conscious, curious, listening to someone talk about biology late at night. The fact that matter can organize itself into something that wonders about its own organization. That's not just biology. That's the deepest mystery there is. So let that settle over you. Let the details blur, if they want to. The names, the dates, the pathways, they'll be there whenever you want to revisit them. For now, just rest in the quiet knowledge that you are, at this very moment, a masterwork of cellular engineering, 4 billion years in the making, running beautifully, effortlessly, while you drift off to sleep.